Hello everyone and welcome back to Chapter 26. We are moving on to the second lecture of Chapter 26, and we're going to start with the first part here, which begins at the renal corpuscle. We're going to talk about filtration, and we're going to talk about regulation that happens here. As we're going to see, the renal corpuscle and all the events that occur here are very, very important because that's the start of forming our filtrate or urine. Alright, so as you can see, we're starting through the nephron and we're going to begin with the renal corpuscle. As we move on to the second lecture, or second part of this, you're going to see that there's the proximal convoluted tubule and the nephron that we'll get to, and then we'll move on to lecture 3 to wrap everything up here. So we are getting into the intense part right now. We're getting into the highly detailed stuff. This is the part where you're going to need to invest a lot more time in comparison to lecture 1. So at this point, make sure you are practicing lecture 1, getting all the anatomy down, because that's the easier part that needs to be well understood to make these lectures make more sense. So definitely invest the time here. As you can see, we're going to be breaking this entire structure down here because this represents a zoomed in version of the renal corpuscle that I don't believe we've seen as of yet. So that's what we're going to be lecturing on. As usual, if you wanted to see the objectives for this lecture, feel free to pause it here. Obviously, since we're on part 1, we're getting through these top 3 objectives, and as you can see, part 2 is basically the proximal convoluted tubule and the nephron loop, where on the first part, we're literally just going to be able to get through the glomerulus or the renal corpuscle, because that is such a key part, because that's the beginning of urine formation. As usual with my first lectures, we know we move along, and so we are following right along with Chapter 26 here, still in our Pearson book here, finishing up the Fundamentals of Anatomy and Physiology by Martini, Nath, and Bartholomew. Alright, let's get into the renal corpuscle. So we're kind of getting into the histology. It's not really histology, it's more on the anatomy side, but we're breaking this down. So that's the main point here. And so again, the renal corpuscle, remember, it's composed of a glomerular capsule and the glomerulus. So here, we actually see the glomerular capsule, right? So this outer region here. And then notice there's space, and so that's going to be called the capsular space. So they're pointing to that, so this open area. And that's where the filtrate, as we form urine, as we start having our hydrostatic pressure force substances out of the blood, they're going to force it into the capsular space here. So definitely need to recognize that. And then this ball right here is going to represent the actual glomerulus, all right? But before we get into that, let's start talking about the glomerular capsule in full detail. So the capsular epithelium, you should know, is simple squamous. So these are all simple squamous cells. And just so we're all clear, this is going to be what we call the glomerular capsule. Okay, so that's it. The outer portion is going to be the capsule. I know the book is linking these two concepts together. We're going to say the glomerular capsule is the outer portion. And then if you notice, that outer portion sort of bends inward and we get these cells that are sitting on top there. Notice they call it the visceral epithelium. We're going to call them podocytes. So we're going to be a little bit different from the book in the fact that we're going to definitely say, hey, this is the glomerular capsule here. And then these cells, which, trust me, I have models that look very similar to this, and they do have these cells on there. And so if I label up and say, what are the cells? They're definitely podocytes. Whereas if I label up the outer portion of that structure, it's definitely the glomerular capsule. And if I follow up either of them, what type of epithelium makes up these cells, or what type of epithelium makes up the structure you just identified, it's going to be simple squamous. So clearly, this structure down here is key to understand. All right, so here they're talking the capsular epithelium is continuous, forming the visceral epithelium. As I said, the visceral epithelium, know them as podocytes. That's what I want you to know. So just know these as podocytes. We're going to keep it a little bit simpler and just say podocytes, but you still should know the podocytes are simple squamous cells. All right, and so they are going to form or wrap around something called the dense layer, and they form something called pedicles. So we're kind of looking down here. All these little extensions are called pedicles, okay? So we'll talk about the dense layer in just a minute, but right now recognize that podocytes produce pedicles. In those pedicles, they're literally cell extensions, and they form something called a filtration slit, which is basically going to enhance filtration. It's basically an accessory structure that increases the ability of filtration or makes filtration a little bit more selective. Okay, so from this, break down this anatomy and make sure you're understanding. We have the this whole structure is the renal corpuscle. We have the outer glomerular capsule. We have the podocytes that are attached to the actual the glomerulus. And so in here you can see this is the glomerulus cell. and then the podocyte sits on the outside. So that's going to be our exchange membrane, or notice our filtration membrane, which we've already seen. And notice there's that word dense layer. So we've seen this already. If you remember, the blood-air barrier was very similar. We had a simple squamous pneumocyte that was attached, or the fused basement membrane, with the capillary, or the endothelium, right? Because that's what capillaries are. The inner lining's the endothelium. And so that's what we had when we talked about the blood-air barrier. Here, this is just called the filtration membrane. It's pretty much a similar setup. We have our fenestrated endothelial cells, so we still have endothelial, but now we have the cells called podocytes, and that's what you should know. We have podocytes. I'm surprised they didn't label up podocytes here. They're focused on the filtration slits, but there's a dense layer that is in between it, and then literally we have our podocytes attached to it. Okay, so that's the major point that you needed to walk away with. Now, if we continue on this, you should note there's something called mesangial cells, and we have two types. You have the intraglomerular mesangial cells. For me personally, those are not ones that I tend to focus on. Okay, and so it's if your instructor talks about it, if you're taking another course and they say, hey, I expect you to know those, that's important that you recognize them. Me, I like focusing on a different set of mesangial cells, and that's these ones out here. All right, notice extraglomerular mesangial cells. And so that's the ones I tend to focus on. And notice they provide support by controlling the lumen diameter, i.e. it impacts the capillary blood flow. So all you need to know about mesangial cells is know that they're structures related to the glomerulus that help control blood flow to and away from the glomerulus. If you have that down, that's about the extent I test on mesangial cells. So mine is kind of just an overview that if you notice those extracellular mesangial cells, and they're pointing here, we follow that goes all the way to the center. Notice that's these orange ones here. Those cells are really going to help control the flow to and away from the glomerulus. So that's the main point. If you have it down, don't get caught up on these mesangial cells. All right, but what's going to regulate their flow is going to be chemicals, either hormones, histamines, things like that are going to regulate flow, as I'm sure we're kind of starting to build the skills towards and starting to really understand that concept of how different ingredients influence dilation versus constriction. Now, glomerular capillaries definitely know they're fenestrated, all right? You need to know that. We're doing large amounts of exchange, but we are not doing cellular exchange. So the albumins have to remain in there. That would be a problem if they escaped, because the albumins, of course, are responsible for creating that blood colloid osmotic pressure, and that's still going to drive osmosis, and it's still going to drive the forcing of substances in specific directions, and we're going to be getting to that. Remember, that's the pooling force when we talk about albumins, the ability to pull substances back in or cause reabsorption. All right, but glomerular capillary plus a dense layer, i.e. essentially the basement membrane, and they're showing that here. We go back here, they called it the dense layer. So I must have taken two images. I think this is an older image. You can see it's from 2015 here, and I think they've just updated the image a little bit better. This is more accurate, so stick with this one because it is talking about fenestrated endothelium plus your basement membrane plus podocytes. Don't worry about the foot processes of podocytes. We're just going to call them the podocytes creating this membrane. Yes, truly speaking, you can see there's little gaps here, and so that's what they're filtering through. They're not actually diffusing through the podocyte, which is why they're calling the membrane the foot processes. But we're going to just stick with the membrane, like I said, the filtration membrane being glomerular capillary plus a dense layer plus your podocytes, okay, or a fused basement membrane. So call it the dense layer or the basement membrane. All right, again, capsular space, I've already pointed out. It's the space where once we form filtrate, it's going to go in here, and as you can see, the next site moves on to the PCT. So this right here starts the PCT, and then if you notice over here, this is actually the distal convoluted tubule, and we're going to talk about why that's relevant. If you notice, there's something called the juxtaglomerular complex. We're going to expand a little bit on that as well. So we're not done with this anatomy, and you can see the renal corpuscle is such a critical structure to fully understand and appreciate because everything starts here. Filtration starts there. All right, so site of glomerular filtration, it's the only location that filtration is going to occur. That's usually a test question that I have. Which of the following areas begins filtration, or where does filtration begin relative to the nephron? Something along those lines, and it's hands down going to be renal corpuscle or glomerulus. All right, blood pressure overall is going to force out about 20% of the plasma and those solutes into the capsular space. So we are doing a huge amount of filtration going on here. Now, note, And this is going to be a little bit weird because usually we're used to things being highly selective when it comes to exchange. But when it comes to filtration, notice it's a highly unselective process. In fact, I often describe filtration as a highly unselective process. Which of the following is a highly unselective process that forces the bulk of solutes and fluids out of the glomerulus? Hands down, you need to know it's glomerular filtration. Now notice, pretty much metabolic waste and excessive ions, those are going to be pushed out. But notice glucose, free fatty acids, certain amino acids, vitamins, useful substance, all are going to enter in the capsular space. But the cells and the plasma proteins like albumins do not pass. That's what I've been stressing when we talked about our various types of capillaries, right? So we have fenestrated here. We don't have sinusoid. Since we do not have sinusoid, cells in these plasma proteins will not pass. All right, the final major structure to note is this juxtaglomerular complex that you can see right here. Now, to be clear, if you notice, there's something called the macula densa. That is part of the DCT that is facing the afferent and efferent arteriole. So that specific region is going to be the macula densa. I do label up the macula densa on the lab exam, okay? Also, you should note there's something called juxtaglomerular cells. And if you look down here, the juxtaglomerular cells, those are part of the afferent arteriole. So they're an extension of the afferent arteriole, and I often label those up. So notice juxtaglomerular cells. All right, so make sure you are able to identify the difference between the macula densa and the juxtaglomerular cells. Those are the main cells that I label up on the lab exams. Now, the function of those cells in general, we're going to call them endocrine cluster that secretes EPO and renin. We know those are the main hormones. Of course, the the kidney also produces calcitriol. But from this region, when we're talking the juxtaglomerular complex, it is strictly erythropoietin and renin, because this area right here, this is where we're going to have our sensing of materials, or sensing of what's in the blood. So that's why EPO and renin, because these are going to be chemoreceptors and pressure receptors, which isn't going to be related to calcitriol at all, or calcitonin in general, right? So work through this. For the lecture exam, I will not single out macula densa and juxtaglomerular cells as an answer. In fact, on the lecture exam, I just expect students to recognize juxtaglomerular complex as being the endocrine cluster that secretes EPO and renin. So lecture and lab are very different on what I expect you to know. Lab, I expect you to know the details. Distal convoluted tubule has the macula densa. On the afferent arteriole, that's where we have juxtaglomerular cells. Combined for the lecture exam, that's just called the juxtaglomerular complex that regulates EPO and renin secretion. So lecture exam, just keep it as juxtaglomerular complex. Lab exam, you have to be able to single those out. And so there's just showing this afferent arteriole, blood comes in, notice this is where we start. This would be the capsular space, so we start forming that. And then we're just moving on into essentially we'd be moving on to the PCT here. And now they're showing you the exchange between the peritubular capillary and the actual tubular network. So that's what we're going to be dealing with when we move onward from here. So right now, this is pretty straightforward because the filtration is coming from the glomerulus. That's the only site of exchange right now. So we're expanding on glomerular filtration. We're going to continue on this concept here. If you notice, we have something happening down here very similar to what we've seen when we talked about capillary exchange. So what we're going to see with filtration, it's going to be 3, or you can consider it 4 factors that contribute to glomerular filtration. And I'll explain why I say 3 or 4 factors depending on how you want to view it. Perspective always matters. But notice here they're showing you the filtration slit. Notice it's a dense layer or the basement membrane. You can use both terms, but here's the endothelial of the glomerulus. Here's the fused basement membrane. Here's your podocyte cells forming the filtration slits. All right, but the first factor is going to be, notice, glomerular hydrostatic pressure. Just like we had hydrostatic pressure when we talked about the capillary region, it's the same thing. We're going to have this pressure that forces substances out of the blood. So this is what's going to generate our ability to have filtration occur. All right. And so notice it's approximately 50 millimeters of mercury. So we're back to that millimeters of mercury. If you remember, well, I'll get to that in just a second. Now notice you also have capsular hydrostatic pressure. Previously, what we had interstitial hydrostatic pressure, and we said that really didn't amount to anything. But notice here, this is the opposite pressure. So down here, there's the 50 millimeters of mercury. Notice here we have approximately 15, so they're showing you this in this little yellow arrow or gold arrow. they're showing that going back in. So there is a force that is pushing solutes back, and that comes out to about 15 millimeters of mercury. Combined, that gives us our net hydrostatic pressure. This factor is why I say it's actually these two components. So this is not a singled out factor, right? This is why I say it's really 3 factors that contribute to it. But if we want to consider this as a separate factor, then it'll be 4. So notice you're going to take the difference between hydrostatic pressures. When we do that, we get 35 millimeters of mercury. Now you take that 35 millimeters of mercury. Remember, we're still looking for our blood colloid osmotic pressure. And so if you remember, and I doubt most of you remember this, it's okay if you don't remember it, but it's constant. That osmotic pressure is created by the albumins, and it should be approximately 25 millimeters of mercury. And so that's the sucking force or the pulling force that are going to pull the fluids back in because of the plasma proteins like albumin sitting here. So now we have this extra 25 also sending or putting force back in this direction. And so the end result, all right, so if we take all of the factors together, we're going to end up with net filtration pressure. So here we had net hydrostatic pressure. When we combine everything together, we get our filtration pressure. And again, net filtration pressure, when it's a positive number, it's telling you that there is a net filtration. If it was negative, it would tell you, hey, no more filtration is going on. So notice we take our glomerular hydrostatic pressure, we subtract it from our capillary hydrostatic pressure. And at this point, if you wanted, you can even just say subtract blood colloid osmotic pressure, and that'll get you your net score. But here, we're getting net hydrostatic pressure of 35. Now you take your net hydrostatic pressure and subtract it from blood colloid. Notice we get 10 millimeters of mercury, so that's where filtration is occurring. All right, so disruptions to our NFP can cause a variety of clinical signs and symptoms, but obviously we want this number to be positive. We want filtration to occur. It doesn't take much to stop filtration. So notice a drop to 40 millimeters of mercury means our kidneys no longer function. So, you know, if your diet goes poor and you're not getting access to water and food like that, that does cause concerns to your kidney. Now, obviously, day to day, sometimes that happens, but if this becomes constant, it can actually lead to some kidney problems and some kidney damage. So it's really important that we have good glomerular hydrostatic pressure, i.e., systemic blood pressure needs to be regulated highly because it would impact this. So that's going to take us to the next concept here, which is glomerular filtration rate here. So when we talk about glomerular filtration rate, the name kind of gives it away. We're talking about how much filtrate are we forming. Now, usually I was focused on a minute when we were talking about like respiratory and we were talking about cardiovascular. But here we're going to just focus on the day, all right? So the amount of filtrate produced each minute is technically the GFR. However, we're going to focus on GFR from the day perspective. And I want you to know it's about 180 liters a day, or to give you a general idea, that's 92-liter bottles of pop being filtered through your kidneys every day. Obviously, it's not pop or soda, it's our blood, but that's a lot of fluid that's being pumped through. This is why I continue to stress that, you know, dialysis and kidney damage, those are really serious things because of how much of a workhorse your kidneys really are. So notice approximately 10 to 20% of the blood plasma is going to enter into that capsular space each minute. So we are really pumping blood through a lot. All right, the way GFR is determined, it's determining by detecting creatinine in your blood for 24 hours. All right, and it should say creatinine. It really doesn't make much of a difference. I'm just giving you the overview of how they would detect this, or how what sort of clinical studies would be done to determine this. It's going to be collection of urine for 24 hours. Now, what I want you to really know is this 100 liters per day. That's what I'm stressing on here. You should know what GFR as a definition is, but know that it's 100 liters a day. Notice about 99% of the filtrate gets reabsorbed. So even though we push out 180 liters a day, you should know that we reabsorb 178 liters, or in general, we form about 2 liters of urine every day. Now, our bladder holds approximately 2 liters, i.e., we're basically filling our bladder every day. Some people can hold it longer than others. I am one of those people that likes going to the bathroom and not feeling that sense to go to the bathroom often. But some people can hold it and they go to the bathroom forever because they, of course, are able to store and hold almost up to 2 liters of urine. All right, so notice less than 1% of the filtrate actually becomes urine. That's the main point. So you should know 180 liters a day become filtrate. Out of those 180 liters, 2 of them actually make it to urine, while the other 178 get reabsorbed, which is going to happen along the nephron. So we're going to be getting to the reabsorption process later. We'll be talking about that in just a bit. Pay attention. Again, 180 liters a day, 178 liters reabsorbed, or 99% of the filtrate gets reabsorbed and only 1% forms urine. All right, GFR does depend on net filtration pressure. So any factor that impacts net filtration pressure, right, is going to impact GFR. We're going to discuss these factors a little bit later. Again, GFR, the main point is just up here, understanding what it is and understanding that we are able to determine kidney health by using GFR. That's the main point. It's not a huge concept that I test on, but you need to understand what it is briefly. All right, so far, main points. Filtration occurs at the glomerular capsule or the renal corpuscle, all right? Or you can say the glomerulus. Either way, I would not have glomerular capsule, renal corpuscle, and glomerulus as an answer choice. It would be one of those. Most likely renal corpuscle or glomerulus is probably the more accurate thing that I would put for filtration. But no, the filtrate ends up dumping into the glomerular capsule, so it's inside the capsular space. Remember, highly unselective. Cells and proteins remain in the blood because we're dealing with fenestrated capillaries, but we are removing all the good and bad stuff. So wastes and toxins get removed, and even our amino acids, our triglycerides, things like that are going to get removed. Fatty acid chains, glucose. All right, this is regulated by our glomerular hydrostatic pressure, our capsular hydrostatic pressure, which ends up forming net hydrostatic pressure. So we take GHP and subtract it from CSHP. Then we also have the blood colloid osmotic pressure that's going to contribute to net filtration pressure. Obviously, from here, this would be a subtraction. I'm just telling you the things that end up contributing to net filtration pressure. So these three factors, or if we consider NHP a factor, that would be those four factors, all contribute to NFP, which means GFR is impacted. Again, main point, GFR is reflecting kidney health. So how do we control the GFR? So glomerular filtration obviously is important for waste excretion, it's important for pH management and regulation, and it's important for regulating blood volumes and blood pressures. But how is this controlled? All right, so this depends on adequate blood flow, all right, and that's what we need. So we're basically going to control blood flow, and that's going to happen via autoregulation, i.e. local blood flow to the organ. Notice we started with autoregulation. What do you think's next? What other things have we talked about that regulate the body? Well, of course, we have hormone regulation, and we have nervous system regulation or autonomic regulation, i.e. the sympathetic division. It's the same exact thing that we just went through with the respiratory system. It's the same thing that we went through the cardiovascular. We always have methods of regulating blood flow. We have hormonal regulations, and then we have neural mechanisms. So we're still doing with the same things. Now, with autoregulation, you have a few things to understand. We're controlling local blood flow to and from the kidney by either constricting or dilating the afferent and the efferent arterioles, and it could even to a lesser extent be the glomerular capillaries, which we are not going to focus on. All right, so here's kind of the question. So if local blood pressure, and again, the key is local because as I've stressed, what happens systemically does not mean it has to happen locally. All right, so if local blood pressure is high, that means GFR would be high. What would happen to slow down this GFR would be the afferent arterioles would end up constricting, and therefore there's less blood flow to the glomerulus, and therefore it puts the GFR in normal limits. So if systemic blood pressure is high and then locally we sense the blood pressure is high, the way the body can adjust its filtration is constrict the afferents. You need to know this. This literally is the only question I can ask because we're not going to say what happens when GFR, this is the only question that I'm going to ask related to how the constriction of afferents is going to occur. On the flip side, if we needed to have an increase in GFR, i.e. our blood pressure systemically might be low and therefore the kidney sense local blood pressure is low as well. If we wanted to keep GFR normal, what can the arterioles do? Well, here we constricted the afferent. So in this case, if we want to get GFR up under low blood pressure conditions, we're going to dilate the afferent, so now we have more blood flow in there, and then we're actually going to constrict the efferent to build pressure. So keep that in mind. These are literally the only two local questions I can ask. If GFR is high, how can the kidney correct it? If the GFR is low, how can the kidney correct it? Okay, so these are your answers. Study this. This is the more detailed question. That's, like I say, these are the sort of questions that you'd expect to separate A and B students from one another. It's really these details down here. All right. The second control is hormonal regulation, and hormonal control of GFR is going to occur via the renin-angiotensin-aldosterone system. So we've already seen this, we've already talked about this, but we didn't call this a system. Now we are starting to call it the renin-angiotensin and aldosterone system. All right, so that's one of the hormones. And then we also have natriuretic peptides. So again, a lot of review here because at this point you should really have these hormones well understood. So the renin system, this causes systemic vasoconstriction of arterioles. We did not talk much about this. We talked about renin converting angiotensinogen, leading to the cascade of aldosterone and ADH release, right? That was our focus. So now we're expanding a little bit here on what renin does. So notice systemic vasoconstriction of arterioles. Again, systemic because we're trying to increase pressure. All right, so the goal is to increase atrial pressure, which ultimately increases blood pressure throughout the body. Also increases GFR as well. So renin release is going to increase systemic pressure and increase local GFR. Where and why is renin released? All right, you should know the specific thing that releases renin. It's no longer acceptable just to say the kidney. What part of the kidney releases this renin? So hopefully you remember renin is released by the juxtaglomerular complex. That's how I will list it on the exam. Remember, this is consisting of the macula densa. Okay, so you need to remember that's part of the DCT. All right, and then the juxtaglomerular cells, which is part of the afferent arteriole. Now, what triggers this to respond is a decrease in blood pressure sense, i.e. the baroreceptor in the kidney is sensing low blood pressure. When that happens, that can trigger the release. So we have a baroreceptor, or systemic pressure is low, or we can actually have sympathetic stimulation. So stimulation of juxtaglomerular cells during sympathetic division, or sympathetic stimulation. So i.e. we get some sort of response that says, hey, increase blood pressure or increase heart rate. We also know that this is going to follow along with it to via the sympathetic division. So an increase in sympathetic activity can cause this renin release. All right. As far as natriuretic peptides go, you should know they cause a decrease in blood pressure by stimulating water loss. So blocking ADH, blocking aldosterone. They dilate the afferent, they constrict the efferent. That causes an increase in GFR, which also causes an increase in urination. So you might want to put natriuretic peptides cause increased urination. And what you would expect is urine that is very dilute. It should end up being mostly water in there, i.e. not many solutes. All right, you can also see there's a decrease in sodium reabsorption and a decrease in blood pressure associated with it. Make sure you're looking at this figure. They're showing you ADH secretion ends up being, in this case for renin, ends up being stimulated. Natriuretic peptides would block this. They would be doing basically the opposite of what you're seeing here. Notice when renin gets released, notice it stimulates tubular sodium and chloride reabsorption and potassium excretion. So we got a lot going on here, but the main points are definitely focused on up in this top region. Really understand these points. We talked about renin throughout the entire semester, so it's important that you recognize it. All right, but to connect everything together, it's kind of right here. Everything gets brought together here. Notice on this, I did not focus much. All right, I did not focus much on the autonomic side. That sympathetic stimulation really was leading to this right here. Stimulation of juxtaglomerular cells. That's about the extent I care about you knowing. We might expand on it a little bit, but notice it's sensing pressure, and so this figure is absolutely important to understand and review. Spend some time there. They're showing you decreased filter pressure, decreased urine production leads to release of renin. That triggers the cascade. That triggers the increase in pressure overall, which then should restore your GFR to normal rates. Use the figure. All right, but that gets us through the first lecture. As you can see, and as I promised, this was going to be the detailed lecture. Part 2, hands down, lecture 2 is so detailed. It's going to continue to be this detailed when we move on to part 2 of lecture 2. But just keep in mind, we're just building off concepts here. Fortunately, I think the second part's a little less intense. This had a lot of anatomy for me to describe, a lot of regulation to describe. We will have some regulation, we will have some anatomy, but the remaining parts are a little bit more basic because we're not going to have much more sensing going on and as much regulation of dilating and constricting blood vessels. Those sort of things aren't going to happen anymore. All right, but we'll get on to the second part to finish up with the PCT, the nephron loop, later. Thank you all for listening. I hope this is coming together all right. As always, if you have questions, please figure out how to get in touch with me. I'll happily respond to you. Other than that, I will see you at part 2 of lecture 2 coming up soon. Take care, everyone. See you next lecture.